Good afternoon, everyone. I am Kirby from MIAtecs. We originally started as a hardware company for smart glasses, but along the way, we decided to shift more to a full solution company. Because from one of our colleagues, Maarten van der Stukken, who is now back to his first big love, research, and our partnership with Bayer, we learned that there was a real need for phenotyping applications. So after diving a bit deeper into it, we started to develop our own data collection app. We developed a web portal where you can configure your observations and an app that works on your cell phones, tablets, and with our smart glasses to run your crop screening sessions. So once you've finished collecting your data, you can go into the app or the web portal and view it, or you can export it. Combining our app with our smart glasses allows you to operate in a hands-free manner, making crop screening a lot less time-consuming. Working like this, by freeing up both hands, so that you can, for example, pull open a crop and check if there are any diseases, and using voice-based data entry results in up to 55% efficiency gain in crop screening data collection. Having the data instantly digital results in up to 20% less human errors, and having the ability to have flexible but structured workflows results in up to 25% increased data consistency. So if you want to learn more about our app and our smart glasses, I invite you all to come talk to us at the upcoming poster session or at our booth that we share with Vito in the exhibition hall. Thank you for your attention.
So hello, I'm Pierre Lejeune from the University of Liège in Belgium. So I'm really glad to be able to present this project, APIC. In French, APIC means something like to be at the right time at the right place. So you might say it's just yet another phenotyping platform. This one has a few specificities. It's meant to be a qualitative phenotyping platform for optimizing high added value production. So how does it do that? So basically, on the upper side, you can see it's a mobile gutter system, 2 stages on top of each other. At one end, there is a hyperspectral imaging station with which we can develop some predictive models. In this example, this is polyphenols in lemon balm. At the bottom left of the slide. On the other side of the system, there's an irrigation station that can perform gutter-wise fertigation, which means that every gutter can receive a different nutrient solution potentially. And the system is a conveyor that cycles the plant at least once a day. And it has also luminaries, LED luminaries with 16 channels that can be pre-programmed to deliver any recipe that you want. And although the luminaries are fixed, the light can sort of follow the gutters as they move in the system. And so we can do also some light optimization experiments with light gradients. And the final goal is to optimize biomass, for example, in medicinal plants, in terms of yield and quality. So if you have further questions, the poster is number 42, which means it will have the answer to everything. Thank you.
So, hi everyone, I'm Fabien Miart. So in the last decade, there has been a lot of development in the field of high-throughput plant phenotyping, especially with robotics and AI. And most of this development have been applied in the field or in indoor platforms. But why this development have been developed in field and indoor platforms? There is still one phenotypic scale which is largely missing from the game. I'm talking about the microscopic scale. So the main reason for that is that the HTMPP and cellular biologists are not talking enough together. So to help bridge this gap between HTMPP and cellular biology, I have launched MIAtecs this year. So let me introduce you with HTMPP for high-throughput microscopy plant phenotyping. And the objectives of MIAtecs is to bring you with new solutions to help you find hidden phenotypic traits from the seedling scale to the subcellular resolution. So the MIAtecs services pipeline is in 4 steps. First, we help you find the best imaging solution and develop your own device. Then we help you find the best microscope that will fit your needs. We also analyze your images or develop image analysis solution that perfectly fit your needs. And finally, we develop advanced analytics tools with graphical unit interface, graph statistics, and visualization. So what are the benefits of HTMPP? You can, with our solution, you can image the shoot and the root part of the seedlings at the same time and compare what is comparable between multiple seedlings. Then you can access the gene expression at the microscopic scale along the seedlings. So you can image, for example, biosensors like pH, calcium, growth, or phytohormones. You can apply chemical treatments very precisely and at low cost, so in an eco-friendly way for your pesticides and chemical products. And you can also, from the early phenotypes, maybe predict what's happened at the plant scale level, so to help you analyze the yield and stress responses. So come to my poster number 41 to discuss a bit in more detail what MIAtecs can do for you and to introduce you with this small microscope here.
So good evening, everyone. So I just want to give a little bit of brief of the work that we do in a Salk Institute. So the Salk Institute program called Harnessing Plant Initiative, which actually try to develop a plant which can have the larger and deeper root system that can store the more carbon in the soil. So we actually wanted to use the plant part to address the climate change. So how this works? Plant takes the carbon dioxide, sends to the carbon forms, and via the root, it stores into the down. So to do that, we have developed quite some multi-modal phenotypic platforms. So these are some of the platforms that we use. These are automated based pipelines, and I'm not going to do on that. What it does is actually it gives more root-related traits up to 1,000 different traits, and we automated all those pipelines to get those data. Using those data, we use the genome-wide SSS mapping and identify the genes, and we also have in a Salk in-house, we created either mutant or overexpressed lines, and those lines are used for the validating those traits using the larger phenotyping modalities. And so we use those modalities just to validate those lines in a larger pod at the flowering stage before that can actually lead to the translation to the field. So you can see one of my colleagues with the root head. So if you want to know more about our work in progress and what the future holds to use the plant to address the climate change, please visit my poster number 43. We will have a fun talk with a coffee. Thank you.
Hello, my name is Daiki Someno. I'm a second-year master student from Japan. I'm interested in developing a system to analyze plant phenotypes using programming, deep learning, and computer vision. In my poster, I will talk about how refining certain steps in a classic and traditional pipeline can improve the analysis of plant phenotypes. My work focuses on 3 key stages. 1, generating mask images. 2nd, using structure from motion and multi-view stereo. And comparing Poisson surface reconstruction with B-spline surface fitting to estimate leaf surface area. These steps are important for precision. For example, the magnification process for mask images impacts the following processes: the types of mask images and the timing of the insertion of them affects the accuracy of 3D reconstruction, and selecting the right surface fitting technique improves the measurement accuracy. Through my study, I want to emphasize the importance of revising each step in the overall pipeline, introducing the comparative study. I'd love to talk more if you are working on something similar. Thank you, and I hope to see you at my poster. Thank you.